Hello and welcome to the IBMS Biopods. I'm Rob.
And this is Becca.
These Biopods will give you an insight into Congress with exclusive interviews, behind-the-scenes chit-chat, and maybe even a drop of science.
So put down your pipettes, move away from the microscope, and get ready for a
Biopod Deep Dive. This episode features two interviews, the first of which Becca was unable to attend, so I'm joined by James.
Hello, I'm James.
And we talked to Dr. Sarah Pitt about her research with snail mucus and antibiotics.
In the second half, we talked to Dr. Rena Richardson about her Congress session entitled How Clean Is Your Mouth?
Dr. Sarah Pitt has also written the cover feature for the December issue of the Biomedical Scientist, so it's a very special month for all those gastropod enthusiasts
and mollusca admirers
who are IBMS members out there and listening today.
On that note, let's slowly slide into this month's Biopod with Dr. Sarah Pitt. Okay, so today we're here with Dr. Sarah Pitt at the University of Brighton.
Sarah, why don't you tell us a little bit about yourself?
Okay, my name is Dr. Sarah Pitt. I'm working here at the University of Brighton as a lecturer. I'm a lecturer in biomedical science practice and also in microbiology. I've worked as a biomedical scientist in microbiology for nearly 30 years now.
Oh, brilliant. So in the current issue of the Biomedical Scientist, we're featuring your work on the cover. So just to jump straight in with that, why snails, Sarah?
Well, I have my husband to thank for this project. He's an invertebrate biologist at another university, and we were talking about what we could offer as undergraduate research project. And he said, there's this thing I'm wondering about, antibiotics in snail mucus. Why don't we look into that? And I resisted it for a couple of years, actually, because I thought, well, I don't know anything about looking after snails. I don't know anything about snails. I'm a virologist. And he tried it with some of his students. And he said he thought that there might be something in it. But when he explained to me what he was actually doing in the laboratory, I said to him, "Oh, you're doing that all wrong." Because I'm the microbiologist in this family, and you're the method you're using. That no wonder it's not working very well. Give it to me, I'll try and sort it out. And so I did it with some undergraduate students the following year, which was 2013-14, I think. And we thought there might be something happening. So I spent that whole summer just in the lab, just playing about with different agars, different ways of preparing the mucus and testing against lots and lots of bacteria, and eventually found that I could get the method to work, but I could only get the mucus to kill one particular species of bacteria. And so that's how we started.
And how did you go about finding those snails? Were you kind of out in the garden at night, little head torch on? What was your snail hunting technique?
At the right time of day and look in the right sort of places. You just find lots of snails. Particularly if it's been raining, they'll all come out. But I did find there was quite a lot around the back of my shed. There was a bit of old flooring, I think, that the previous owners of the house had left, and underneath it, I picked it up and I lifted it up, and there was tons of snails underneath it. So they all came indoors for a nice life of being warm and secure and being regularly fed. I'm sure they loved it.
And for those listeners like me who don't know a lot about snails, in a typical garden, do you find lots of different types of snails, or is there one predominant species?
There's one predominant species called the brown garden snail, which is the one that we'll be working on. But you do actually get other smaller ones, the sepia species, which they're much tinier, and their shells are they're kind of more coiled, and they're more different colours, and they're smaller. Those are the two main ones that we found. But the brown garden snail is the one that we've been concentrating on. We've tried sepia, but the mucus is, because they're small, it's quite difficult to get mucus out of them, and we didn't find that it worked very well anyway.
And has it changed the way you think about snails? I don't think of them as particularly nice or pleasant things. I think of them as things where I'm going to put the bins out and I've accidentally trodden on one and there's a horrible squelch and I wish I'd put my shoes on, but I didn't. How do you think about snails?
Yes, as I say, I didn't know anything about snails and was slightly reluctant to get involved with them at the beginning. But now, one of the things that you do have to do is you have to handle them. And if you have a snail on your hand, just wiggle, it's delightful. It makes you feel really happy. It's just kind of makes you feel the snails, the little antennae come out because they want to see what's going on, and they'll look at you and they'll just carry on walking. You know, it almost looks as though they're coming out to say hello to you, which obviously I know they're not, but they're very comfortable. The ones, particularly the ones we have in the laboratory, are very comfortable with being handled, and so they actually quite enjoy it. But it really is a delight to have an animal on your hand that's just interacting with you, and in that moment, it's just you and the animal, and there's something quite special about that. I've completely changed my mind about snails.
And this is possibly a stupid question, but do they have emotions? Do snails have emotions? Can they show fear and joy?
Well, they can. I don't know whether you'd go as far as saying emotions, but obviously, if they're afraid, they will curl up inside their shells and produce the defensive mucus, which we're actually interested in. So that can be a bit of a problem with trying to collect the mucus because they're not afraid of me.
So do you have to try and scare them?
I have to try and scare them.
What are your scaring techniques?
Poking them with a cotton wool bud until they get upset.
And talk us through the process of, so you poked it with a cotton wool bud, it's secreted the slime. How do you get from that point to potentially having an antibiotic? There seems like a massive swathe of things in between.
There are a lot of things in between. So what we do is we collect that mucus. I gather from my research students that actually the latest method is scrape it all off with a scalpel. But before I was trying to harvest it with a plastic pipette, and it takes a really long time.
I bet.
We then stick it in the cold room overnight because we found that 4 degrees was helpful. We tried various temperatures. We found 4 degrees works nicely with some glass beads and then in a shaker to kind of try and get the things we're interested in out of all the gloopy bit of the protein because it's very sticky and it's very viscous. And then we dilute it in phosphate buffered saline and then centrifuge it down the next day. And then we can put it through a series of filters with different sizes of the membrane to actually try and get rid of some of the impurities and to try and get down to the individual sort of bands where the proteins might be. And then we have that. So that's as far as we've got really. In terms of making it into an actual antibiotic, we just, there's a lot more steps that we've got to go to. So our more recent work, we've actually been able to identify 3 key proteins that we're actually interested in. But what we're trying to do is express them artificially in an Escherichia coli vector to try and make lots and lots of the protein outside of all the gloopiness of the mucus.
Yes.
So we can actually start doing things like minimum inhibitory concentrations for that protein and start characterizing the protein in more sort of biochemical detail so that a pharmaceutical company might actually be interested in it. I'm sure they're not. I'm sure if we just give them a load of mucus and say, here, get your antibiotic out of that, they're not going to be that keen. But if we say, here's our protein, we definitely know it's the thing that's working. And also if we can get that protein on its own, we can work out how does it work against the bacteria, because that's the thing we don't really know. It seems to specifically work against Pseudomonas aeruginosa, but we don't know why that might be or how it's actually doing that. So we need more information about that.
And is this an evolutionary thing that snails have kind of developed to protect themselves against something?
Well, it does appear to be. There are 2 really intriguing things that I found out when in the analysis of the results that we've got. The first is that when I looked at just Googled snail care, I actually found that there's guidance if you're looking after a pet snail, including if you take garden snails in and keep them as pets, that Pseudomonas is a particular risk that snail care owners snail care experts are aware of. So I think that's quite interesting. And the other thing that interests me is the fact that Pseudomonas aeruginosa is an important pathogen in people with cystic fibrosis, and it performs biofilms in the mucus in the lungs of people with cystic fibrosis. And obviously it's forming potentially the mucus of the snails potentially could have a biofilm. So they might want to protect against Pseudomonas specifically for the same sort of reasons and in the same sort of way. So potentially the answer to your question is yes, I think so.
And is it something that we would find in other creatures that kind of interact with the soil microbiome, like worms, centipedes, slugs?
Yeah, we think so. We haven't. It's quite We've tried slugs. We've tried looking at slugs, but the trouble with slugs is that when they produce their mucus, because they don't have shells, their protective mechanism is for the mucus to turn really hard almost as soon as it's being produced to kind of encase around the slug as a protective thing. So snail mucus is gloopy and viscous, but it's still sort of more or less liquid. Snail mucus goes into a really hard lump, and we have actually not been able to so far find a method that allows us to actually extract to make it into a liquid enough form that we can actually extract anything from it and actually do any experiments on it. We've tried different chemical methods of sort of maybe on thickening it, but then it turned out that the chemicals, the method that my student used, the method that was that she found that was advocated, turned out that it had a chemical in it which had antimicrobial properties anyway. So we couldn't work out whether it was the mucus or it was the chemical that was killing the bacteria, but everything died. So we were kind of back to the drawing board on that one. But there is research for things like sea slugs and other sorts of mollusks where they're looking at the mucus and finding them it might have antimicrobial properties. It's still it's kind of a new area of research actually, really across the board, but potentially yes.
You mentioned your students there. How do they feel about the snails? Have you had any that have hated snails?
Most of them start off thinking they're a bit creeped out by it, but in the end they end up loving them like I do.
Okay. And do you think the kind of natural world is a bit of an untapped area where we're going to see increasing research into kind of antibiotics with the rise in antimicrobial resistance?
I think it could be. I think it could be a place that people could be looking at a little bit more. I think obviously we've found some key antibiotics, and then what people decided to do was just tweak the chemical structures of the key antibiotics, which we know would work. And I think some of the other avenues of research, for example, like phage therapy and investigating natural remedies, have sort of been put to one side a little bit because we believed in the drugs that we already had. And quite rightly, they were amazing. But now that resistance is becoming a problem, people are having to be a little bit creative and going back to perhaps things which were thought of 100 years ago and shelved because penicillin was better and more available and worked well and was the thing. But now perhaps we need to go back and have a look at some other.
And how big a problem do you think antibiotic resistance is? Is that something that is... There's a lot of coverage in the media. Is it something that's going to have a tangible impact in our lifetimes? Is it already having a big impact? Or do you think it's something where we'll be able to find more antibiotics over the coming years? It doesn't look very promising at the moment.
It doesn't. The World Health Organization does consider it to be a major public health problem. And resistance is growing, and there are now strains of certain species of bacteria that are resistant to every antibiotic that we could possibly use to treat with it. So if you get infected with that particular strain, then you may well not be able to get the treatment that you need. And there's only a few strains of a few species so far, but the fact that we got any at all is a serious worry, I think. We could pull it back from the brink, I think, if we start using antibiotics a bit smarter. And so don't take antibiotics if you don't need them. If you've got a viral infection, don't demand antibiotics from your doctor. Also finish your course of antibiotics. People often get halfway through the 7-day course, think, I feel better now. And then don't finish the full course. And then that allows the bacteria to kind of maybe come back and regrow and develop it encourages the development of resistance. So people have got to use them a bit better. But still, I'm not sure whether we'll really get back to how it used to be in the beginning of penicillin, where it cured everything.
Is it possible for those resistant strains to die out, or once they're here, are they here for good?
Well, if you found something that killed those resistant strains, then that genetic line would die out. But the fact that they're resistant, most of them are resistant to more than one type of antibiotic, is actually quite difficult to find something that would kill that whole line off. And also because of the way our modern world is, those strains have travelled all across the world. And so actually you'd have to find a drug that would definitely kill them all dead and definitely find every single isolate of every single version of that strain in every single country of the world in order to stop it spreading. So it's actually quite difficult to do that, I would think.
Oh, definitely. But going back to something which will be helpful, your research, what happens next with your research, Sarah?
Well, as I say, we've got these 3 proteins that we're interested in, and I think there are going to be some more as well. We're trying to make it artificially in the lab. It's not working tremendously well, but the other thing I'm going to do after Christmas, I hope, I've got some funding for it now, is try and crystallise the protein so we can actually see what it looks like. That might start giving you some clues about how it might work against bacteria as well. And then with some more, I've got a lot of clinical isolates now from various different sorts of patients, and I want to just test my mucus against all of them and see whether it works against every single type of, so far it seems to work against every single type of Pseudomonas aeruginosa we've tested it against. If I can get an extensive range of isolates, then, and we can show that it works against most of them, then a drug company might be interested in taking that further, because obviously that we'll get to the point where it's the limits of my ability to do more with it.
And what happened, like, how do you go from that step where you've got something, you want to give it to a pharmaceutical company? Do you kind of say, we've got this is for the good of the world, go and develop it? How does that process work, and who gives who money?
I think if I just did that, the university might have something to say about it. There are processes and procedures that you would go to by actually, sometimes people patent things for a specific use. So it's tricky because I haven't invented it. I've just discovered something that was already there.
Yes.
But you can patent things for a specific use, and drug companies are apparently interested in that. And then the university has sort of things about intellectual property rights according to the information that you've actually managed to glean in your lab research, and so on. So it's not. There are people who I know I can talk to, who I have already talked to within the institution about that sort of procedure. I'm quite keen to, if it can turn into a new antibiotic, then I'm quite keen to get there as easily and as possible. But you have to be a little bit careful, so I get that. Of course.
And what type of timescales are we looking at? If everything went incredibly well as possible with your research, at what point could a new antibiotic be prescribed that's based on your work, do you think?
Well, we're looking sort of 5 to 10 years minimum, I would think. It depends on what anyone wants to do with it. So you can buy face cream in health food shops and some sort of cosmetic things, which apparently this cream has got snail mucus in it, but it's just general snail mucus. It's not. They haven't actually taken out particular elements of the mucus. They've just put whole mucus in there, presumably cleaned it up a little bit, and put it in a cream. So the fact that you can put whole snail mucus in a cream, and that is something that's been through safety testing and is being sold to people over the counter as a useful thing, suggests to me that we might be able to put our snail mucus protein into a cream, because we know that it works against, we know that Pseudomonas is a cause of chronic infection in wound infections, and we have actually tested our mucus against bacteria that have been taken from chronic wound infection, so we know it works. So it could potentially be something that you could turn into a cream to put on somebody's wound dressing, which is a thing that people are starting to do anyway. So kind of a more targeted treatment within your actual wound dressing to help clean your wound up more quickly. So I don't think it would be too difficult to do that. to go any further and make it into a sort of a tablet that you take might take a bit longer, because you've got to do the formulation from it, and then you've got to do all the clinical trials and safety testing. So I'm sort of quite hopeful for the wound cream, at least.
And snail facials. I remember reading a few years ago in all the newspapers about this new craze at some high-end salons where they put snails on your face, let them have a little wander around on there. Is that beneficial, and why are people doing that? And have you ever been tempted or done it yourself?
I wouldn't advise that,
No?
because you don't know where the snails have been, really. I mean, what we found is that they have lots of bacteria and things that I mentioned earlier on, that we put our mucus through a series of filters to get rid of impurities. If you just put a snail that hasn't, even if it's been in a reasonably clean environment, it's still got other bacteria and other impurities on the outside of its body, which you're potentially exposing your skin to. So I wouldn't recommend it, no.
So a warning for all listeners at home, do not put snails on yourself if you're trying to make yourself better or improve your beauty, I suppose.
You can get a snail face mask
Can you?
and things like that. They'd be okay. If it's being kind of prepared into some kind of potion, fine, but I wouldn't
Don't get
a
for
live
snails and
snail
do it
and
yourself.
put it on yourself. No, I wouldn't recommend that. No.
And finally, before I pass you over to James for the quickfire round, have you ever eaten snails, and would you ever be willing to now after your research?
No, I've never eaten them. I've never really fancied it, and now I definitely wouldn't. As I've got older, I'm becoming increasingly more and more vegetarian, and there's very little meat that I'll actually eat. Every time I fall in love with a certain animal, then I won't eat the meat of said animal.
Yeah.
So definitely, snails will definitely
Off the menu.
Definitely off the menu.
Brilliant. I'm going to pass you over to James for the quickfire round.
Brilliant insight, thank you. Okay, Sarah, this is your quick fire round. Are you ready? What's your favourite pathogen?
Oh, that's easy. Dengue virus.
What's your favourite piece of lab equipment?
I'd say a microscope, because it's quite versatile. You can use it for lots of different things.
What was your favourite thing about Congress?
Oh, meeting people. There are people I only ever see at Congress, but I always bump into them at Congress, and I love that.
Okay. Describe your job in just 3 words.
Varied, stimulating, and tiring.
What's the best piece of advice you've ever been given?
I've been given lots of very good advice by a lot of brilliant people along the way, but I think the one that crops up quite often is believe in yourself. The fact that people have to keep saying it to me suggests perhaps I'm not taking the advice as I should do.
And lastly, who's your science hero?
Well, the people who trained me in my very first job as a biomedical scientist are all really amazing people, and they're people I admire and respect. But I'd say as a scientist, as a microbiologist, I'd have to say Louis Pasteur.
Brilliant stuff, thank you. And I shall pass you back to Rob now for your overrated, underrated and correctly rated section.
Thank you very much. So, Sarah, team building exercises, are they overrated, underrated or correctly rated?
I think they're overrated. I don't think they necessarily achieve what they're set out to achieve at all.
Have you been involved in many, or have you been lucky enough to avoid them?
Not very many, but the ones I've been in haven't really helped the team to be built better.
Lab coats, overrated, underrated or correctly rated?
Well, I'd hope that they were correctly rated, but actually I suspect probably they're underrated. But they're a really, really important part of safety. And as a microbiologist, I'm very keen on people wearing their lab coats, wearing them done up properly, and using them effectively. And so they're probably underrated.
Any renegade students that you have to tell off for incorrectly wearing a lab coat?
Oh yes, all the time. They don't. As they come into my class, I'm always telling them, do your lab coat up properly before you walk through the laboratory. That's one of my. They could probably put that on a tape and just press the button.
Nice.
For every lab class. Yes.
Socialising with colleagues.
I think that's underrated. I think people have very much more disparate lives than they used to have, and in the university, people are doing different things. That here in my department, we're in different bits of different buildings. Actually, we don't see each other very much. But then everyone sort of goes home to their families, and in diagnostic labs now, everyone's doing different shifts, so we don't really see each other. And the effort to get together and to go out for a drink on a Friday afternoon, for example, or a meal, is sort of, it's harder to get everybody together because we're not all together to go, oh, shall we just go for lunch? And so it's really important that you do that, just sort of just getting to know each other and just having a chat about even problems at work can sometimes be solved over a sandwich. But so it's definitely underrated because we just don't make the effort to do it as much as we should do.
Good answer. And birthday cards and leaving cards and the little messages that you have to leave in them for colleagues. What about those? Overrated, underrated, or correctly rated?
For colleagues, I think they're overrated because it's very difficult to think of something that you really want to say to them. Hopefully, whatever it is you really want to say to them, you've said it to them. And so in the card, you've only got a little bit of space. Everyone else is going to read it. So you probably just say, good luck, best wishes, keep in touch.
Have you got a go-to message? Your typical one where it's like, I know this person a little bit, not great, this is my message.
Well, good luck for the future and best wishes.
Yeah, nice. Social media in the workplace and the use of social media to promote the workplace.
I don't really use social media that much. I look on other people's social media pages, but I don't have one. So I think probably underrated. I think it probably is a good way of getting your message out to a lot of people. But I think perhaps those of us who should be doing the messaging haven't really caught up with the fact that it's so widely accessed by people, and not just the people that you know about or within your profession, but in the wider world as well. Lots of people are on Twitter, for example, or Instagram or Facebook. You can get a message to a lot of people that way, and I'm only just catching up with that myself, so probably underrated. Underrated.
And the final one, night shifts, overrated, underrated, or correctly rated?
Well, it's a long time since I've done a night shift. Unless it's a self-imposed one because I'm kind of working late in the lab. In today's diagnostic service, they are really important way of keeping the service running. It's a requirement for many labs to actually have a night shift. So I suppose they're probably correctly rated in the sense that everybody knows we have to do them, but hardly any. Some people like night shifts, but mostly people don't like them. And so we know they have to be done, but we don't really like them, which is correct rating, really.
And if you're working back in diagnostics now, how do you think you would get on with night shifts?
I'm a morning person, so I don't think I'd get on with them terribly well. Really.
Not for you.
Not for me.
Brilliant. Sarah, thank you so much for your time. It's really appreciated.
Thank you.
So thanks to Dr. Sarah Pitt.
Now on to our next interviewee, Dr. Rena Richardson.
So we're welcoming Rena Richardson with us today. Rena, welcome to the IBMS Biopod. Thank you for having us. And would you like to just give our listeners a bit of an introduction about what you are, what you do, your job title?
So I'm a medical mycologist working for the Manchester University NHS Foundation Trust, but I'm also a senior lecturer in infectious diseases for the University of Manchester. I'm lead for the infectious diseases team, and I do quite a lot of research, mainly on fungi, but also other things.
So tell us a bit about your background, Rena, because it's quite unusual, and your education and your kind of path into the role that you've got now.
So, if you like to study a lot and do a lot of exams, you do what I've done. So my first degree was dentistry, and that's why the topic of today was the link of basically dental health and oral infections and impact on general health. I moved on from dentistry, did a PhD in immunology, and then started training in infectious diseases, microbiology and infectious diseases, and that's my current role.
And in today's talk, you were talking about the microflora, oral microflora. Can you explain a bit about what that is? Because I'm not sure how many of our listeners will know.
Well, it's the normal bugs that live in your mouth that consist of bacteria, fungi, viruses, not that many parasites, but possibly those as well. So it's the mixture of them that mouth is their home.
And how many are there? How many, if we say, looking at bacteria, how many types of bacteria would there be in the typical person's mouth?
Well, we normally say it's about 500 to 600 species in general have been cultured in human mouth. Molecular methods maybe find 10 times that. But in one individual, it's a couple of hundred species. And in one corner of each specific area in the mouth, like the gingival pocket or the tongue surface or the cheek surface, it's only maybe less than 100 species.
So are they very localized then? You would get a specific bacteria on the tongue, another one at the...
Yes, they are, because they have different growth requirements, and those areas are very different. You know, on the tongue, they have to tolerate being brushed and touched all the time, whereas in the gingival pocket, they can just hide and sit quietly. Nothing is going to touch them unless you use your toothpick and floss and so forth.
Yeah. And I think in your talk, you mentioned that you get some sociable bacteria and some unsociable bacteria, which I didn't know about. Can you explain the difference between those?
So, yeah, some bacteria are found far more in the dental oral biofilms. And biofilm is a social community of bacteria where they share responsibilities and almost work as a multicellular organism. Biofilm is a lifestyle between unicellular microorganisms and multicellular organisms. And so some bacteria and fungi definitely prefer the social lifestyle and they live in biofilms, whereas others are less sociable and they like to hang around and on their own and disseminate and are free agents more.
And are they good? Are they bad? Like, what's the percentage of good to bad bacteria, if we can class it that way?
Impossible to say. So the mixture of healthy oral cavity, the mixture of bacteria and healthy oral cavity is varied between individuals, and it is vast. There are some species that are associated with dental infections, and it's more the opportunity how the conditions change for them to take over rather than that the bugs wouldn't be there. There's most of the pathogens that we find in dental disease and infection actually are present in the healthy mouth as well. They just find the opportunity to take over, and the situation or the circumstances are favorable for them to overgrow.
And does the population change according to kind of time of day and what you've done? Like, say I've just got up in the morning, brushed my teeth. Am I going to have less bacteria and fungi in my mouth then than I will 3 hours later after I've just eaten a snack?
Yes, environmental things do affect, and we know that the microbiome of people who eat, so there are lots of natural antimicrobials that we are exposed to. Cinnamon, some spices, garlic, they are antimicrobial. So if you eat a sort of Indian curry kind of diet, your microflora is different from, toast and jam diet. Obviously, any other thing that we know that the hospital environment radically changes what bacteria you can find in the mouth. Is that partially the antibiotic pressure in hospital ICU, or is it just that you may be with your mouth open there, being intubated with the tube in your throat, and the dry environment then changes everything. Combinations of things.
Yeah. So if there was going to be an ideal diet, and I'm not saying, obviously the ideal diet for to have the right kind of bacteria in your mouth versus a healthy lifestyle probably doesn't quite add up. But what would be the best things you could possibly eat if you were just focusing on, I want to keep my mouth really healthy and have the right kind of bacteria?
For the healthy mouth, the key is regular, thorough oral hygiene once daily.
I thought you were going to say just have a curry all the time.
No, sorry. It's the boring old, it's like hand hygiene, hospital infection control. It's the same thing. Infection control in the mouth is all about oral hygiene. So proper brushing and flossing twice a day is the cornerstone of everything. Mouthwashes are not needed. Often mouthwashes have all sorts of active ingredients that are only active against part of the oral flora, and they can distort the balance between different members of the flora, which is generally not a good idea.
So they can be actively unhealthy, mouthwashes.
And for example, promote thrush in the oral cavity if they kill everything except for the yeasts. So besides that, about what's the healthy diet, it really doesn't matter as long as you have 3 to 4 hours between your meals.
Why 3 to 4 hours between meals?
Because that's the time it takes for the oral cavity to recover from the previous meal.
Yeah.
So it's saliva neutralizes, saliva repairs the damage that the bacteria do on your teeth when they start producing acids, when they get sugars. They don't need purified sugar to produce acids. They are older than humans, so they live on fructose and lactose and all those things. So so any normal meal with some calories in it is good enough for your oral bacteria. And for a couple of hours, they do produce all sorts of things that will cause damage. But another couple of hours, the mouth and the saliva will fix itself.
And if I had a snack in between 2 meals, so I've had my lunch, it's 4 o'clock, I fancy a snack, and then I'm going to have my dinner at 6. Should I avoid the snack?
Oh, definitely, because that is the dip back to the bad state, the erosive state of your mouth.
What about just like a chocolate, one chocolate?
Oh,
that's okay, right?
Nope.
Not even.
The smallest amount. It's quite the smallest amount of any additional energy for the bugs it triggers it. So it's said that if you have a proper dollop of milk in your tea, that's enough.
Yeah, that's enough. Okay.
But milk with no sugar, no milk, tea with no sugar or milk or anything, that doesn't trigger normally.
Yeah.
That sort of, or sugar-free drinks or water. Yeah. Obviously.
And in your talk, you mentioned that fungi in the mouth. I think people might be surprised to know they've got that. How does that happen?
It's just normal things. So there are both yeasts and molds, filamentous fungi in the mouth. Yeasts are normally from your own colonization. We all carry yeast. Some of them are from diet, baker's yeast. Many cheeses are produced with fungi. Blue cheese is blue because of blue mold, and so forth. So microbes are used in food production, so we get it from there. Molds, on the other hand, disseminate as normal by producing fungal spores, and we inhale those spores whenever we are outside in the autumn, walking in amongst the leaves. That lifts up, mold spores that eat the debris. Yeah.
And they just stay in your mouth once they're there?
Who knows? Yeah. There are very few studies following up the consistency of that. And yeast, yes, those fungi that are your colonizers, definitely they hang around and they are very sort of permanent colonization. But but molds, who knows?
Yeah. And with the bacteria in your mouth, can do things move from your mouth into your bloodstream? And how does that process work? Sorry, we're just recording something. Sorry. What was my question? I'll say it again. Yeah. So how do bacteria make their way from your mouth? I understand they can get into your bloodstream. Is that right?
They can do so, and they do, and it's very common. So that's why the immune system is kept alert all the time. So when you eat anything, chew something, you clean your teeth or bite your tongue or lip. There is always a portal of entry for few bacteria or fungi into your bloodstream. But the immune system clears it. It's not a problem.
Yeah.
It becomes a problem if the amount of bacteria getting into your bloodstream is bigger than what the tolerance or capacity of your immune system is. And then you can start getting spread of infection, disseminated infection.
Yeah. And leading on to a final question from this section, I think you mentioned that bad mouth hygiene can potentially be fatal. Am I over-exaggerating that? Possibly.
Yes, it can be. So it's a bad combination to have, pus oozing pockets, and then potentially something happens in the mouth. You get a massive amount of bugs in your bloodstream, and that can lead to endocarditis, infection of your heart, or it can go to your brain and so forth. So there are numerous cases like that. It's rare, but it's more common in patients who have some underlying conditions. So if you're treated with immunosuppressants, like for rheumatoid diseases, or you're undergoing cancer therapies or anything like that, when your immune system is less capable of managing those transient bacterial exposures, obviously then your oral hygiene should be super effective and good.
So one more final question, actually, just so we don't end on too much of a depressing note before we move on to the quick fire. What should people's ideal kind of brushing routine be? Just twice a day, after every meal? How long? And have you got a favourite type of toothpaste?
Oh, God. So we worry about the acid production by the bacteria when they get any energy. The acid production starts when you eat. So the best way to reduce the risk for acid production is to clean your mouth before you eat, because then the number of bacteria capable of producing acid is minimized. And the acid attack, or whatever you want to call it, is sort of slightly controlled. Brushing immediately after meal, on the other hand, is a bad idea, because that's when the tooth surface is at its softest, because the acid have etched the surface soft and the saliva hasn't repaired it yet back to its strength. And then if you brush, then you just basically file the tooth surface off and it will never recover. Yeah. Toothpaste, whatever, as long as it has fluorides in it, I don't mind.
Brilliant. So I'm going to pass you over to Becca for the quick fire round.
Excellent. So these are just quite light-hearted questions. So we'll kick off with, what is your favourite microbe?
Candida albicans.
Why?
It is so versatile. It's a really interesting. It can colonize you. You can carry it, and then with multiple things happening in your body, it all in the sun wakes up and causes disease. It reacts to your hormonal balance. Vaginal thrush is estrogen driven. It reacts to immunosuppressants. It's just fascinating.
It sounds scary. Excellent. Can you describe your job in 3 words?
Challenging, really exciting, and in some ways satisfying.
Oh, that's nice to hear. Good job satisfaction.
Yeah.
Excellent. Do you have a science hero?
What do you mean by that?
So like your
No.
favourite scientist?
Oh, no. You can always say Einstein, but I liked his hairstyle.
That's a good enough answer for me. What's the best piece of advice you've ever been given? To brush your teeth?
No. I think it had to. This is a bit silly, but it has to do that I'm quite sort of active when we go to conferences. I want to be involved, and I ask questions, and I talk. A sociable person. And one supervisor told me once that, Rina, you don't have to stand up all the time.
So that's probably a metaphor for your life as well. Probably sit down for a minute.
I've calmed down since.
Yes. Good. Do you have a favourite piece of lab equipment?
Microscope.
You're not the first one to say that, actually. Excellent. And finally, what is your favourite thing about Congress?
The mix of people and specialties and, just the mix.
Socialising and networking, yeah.
Yeah, but also topics and too often we go to microbiology meeting or clinical chemistry meeting, but now you can pop into slightly outside your core topic and see what's going on the other side.
Yeah, that must be nice actually to see what else other scientists are doing. Excellent, thank you. I'm going to pass you back over to Rob for our final feature.
Okay, so this is overrated, underrated, correctly rated. And the first one is team building exercises. Are they overrated, underrated, or correctly rated?
I think they're correctly rated, generally.
You think people rate them well?
It depends who does them and how you do them. But if they're done well professionally, not sort of ad hoc, within teams, somebody read a book kind of thing, but properly professionally done, I found them, I've enjoyed them, and as a on the receiving end, and I think they are good. Yeah.
Lab coats. That was a telling noise.
Oh, that's a tricky one. I think there again, they are appropriately probably in balance. They are fine. Some ways a bit OTT for some things to step 1 meter into the lab. You have to wear a lab coat and you only are talking and asking a question and walking out. Sometimes a bit frustrating. And is that sort of playing around with the coat, creating, shedding of everything that is on the dirty coat? Is that safer than not disturbing the air and the bugs? But on the other hand, I think it's in the past people just did bench work basically, wearing nothing. I'm not pro that. So.
Socializing with colleagues.
I think it's equally depends how you do it. But in my own life, I think it's sort of in balance. Is it overrated generally? No. Underrated? No. It has its sides. You have to have life outside work, of course. It can't be your only social circle. But on the other hand, work is an important part of your life, and you have to be able to talk about your work with people who understand what you're talking about. So most of us are not married to somebody who works equally in your lab. So you can't, who else would listen?
Yeah. And writing messages in birthday and leaving cards for colleagues. Overrated, underrated, or correctly rated?
I think it's correctly rated. I've changed jobs in my life, and I do keep those cards, and I find it interesting to think, not necessarily the wording of the messages, but it's just nice to go back and see, oh yes, that was the team I worked with. And
have you got a go-to message for if you don't know the person very well, but you feel like you're going to sign it?
No.
All the best, Rena. A little kiss on the end. Or?
I often, my role is to be the lead for the service, so I often thank for being a team player and contribution to the whole service.
And social media at the workplace.
Well, I do clinical work as well, and I think that's just not flatly doable. It doesn't go together. So you're not taking pictures in clinic and saying exciting patients today. No. In the labs, it can be. It depends a little bit what we're doing. If you're talking about private things and
I suppose promoting what is happening in the lab through social media when people kind of
I think it's not too bad. I think otherwise we all live in our little bubbles, and I think sharing what you do is probably a better part of social media rather than what your new hairstyle is. So some professional social media actually is more into my liking.
Yeah, okay. And finally, night shifts, overrated, underrated, or correctly rated?
I think they're overrated. Definitely.
Have you had to do night shifts yourself?
Oh, yes, of course.
Yeah.
I don't do at the moment, but in the past, of course, yeah. And of course, as a clinician, somebody has to cover the wards in the night, and of course, that's different. But for laboratory people, 24/7 services, I'm not sure we actually know what we're doing with that. There's quite a lot of lack of understanding that you have to basically increase your workforce by 1/3 to be able to do it. And do we really need that? And and the impact on quality of life, family life and everything else, is that the right balance? So I think the benefits are probably overvalued in contrast to the cost on quality of life.
Yeah, brilliant. Rena, that is everything. Thank you so much for joining us, and thank you for your time.
Sure.
Thanks for listening. We hope you enjoyed the episode, and don't forget, this can be used for your CPD.
See you next month for another Biopod. This is Becca.
And Rob,
Signing off. Bye.